I hope the week on using labs to study molecular dynamics was useful, and that you were able to kind of grasp the concepts, ask your questions. Today, we're tackling a really quite different topic, actually. So I wasn't planning on going back on the concepts of molecular dynamics because we had the labs. So I just want here to introduce a really quite different topic, which is bioinformatics. And I'll say the book that you're studying, Finkelstein, is really not updated on this topic, but this is a technique or an ensemble of technique that is used a lot in research and to study biomolecules. So this is why we introduce it, but it means that basically the contents of this topic will be kind of the slides and what I say in here. There's not much in the book about it. So I guess bioinformatics is a kind of an obscure type of word. What does it mean exactly? I guess we recognize bio, which means we're going to study biomolecules. And informatics is not a word that is really used as itself, but it's a kind of synonym of data science, information theory, right? Kind of just using computers to study the data and then apply this to biological problems. So it should really be thought of as not different from biophysics, but that it's complementary and that you kind of combine information from this kind of branch that I will describe today for 2 hours with what you have seen up until now and what you will keep seeing in the rest of the class. Yes? So really what we need to be thinking about here is this basic relationship between sequence, structure, and function. And I think Eric introduced this in quite a lot of detail already in your first lectures. This is the idea that for protein biophysics, the sequence of amino acid encodes, or let's say, the sequence of the nucleic acids encodes the sequence of the protein, which encodes the structure, which then gives rise to function. And really what we're going to be talking about today is that by just studying the sequence that is gathered in many large databases, we can learn things about structure and function. So I'll mention today several databases that are useful, and I'm going to basically teach you how we can compare sequences both in terms of genes and in terms of protein. And we'll learn how we can infer things about these two other things, structure and function, from the sequence. So this whole lecture is about finding connections between these three layers. Yes? So you might view this for the moment as a more intellectual problem. So I guess you study science in some part to just be able to understand how nature works. And in and of itself, it's kind of intriguing that DNA encodes RNA that encodes a protein sequence, which is the basis for structural arrangement and then function. But of course, this is not the only aim. As always, when you understand something in terms of science, then you can develop some technology and you'll find some practical applications. So understanding this is quite crucial to make predictions, and with predictions, we can possibly do useful things. So the basis of creating these big databases of sequence is DNA sequencing, and you don't need to know much about this other than it can be done using such a machine. There's been a very big effort in the last decades to map DNA sequences. It became much faster and cheaper. So if you think about the Human Genome Project, which was completed already a while ago now, that used to cost $2.7 billion. And today, to sequence an entire genome costs $1,000. So imagine how cheap it is. And actually, where I work at SciLife Lab, we have several machines that are able to sequence any type of DNA, including viruses, but not limited to. Using this kind of machine. So I myself don't know too much about this technique, but basically the idea is that in DNA you have complementary base pairs, right? You have 4 types of nucleic acids, A, T, G, C, and A and T are complementary, and C and G are complementary. So if you have a primary sequence that you're trying to determine, having complementary pairs that react with their complementary pair and trigger some chemical reaction that is different for each of those, then you can detect the sequence. So this basically says that with red pairs green, and green generates the release of 1 proton, which can be detected then in this detector. Whereas yellow, which is complementary to blue, generates the production of 2 protons, and so this is detected differently. And this can be done extremely fast in an automated way. And this means that we can sequence single-stranded DNAs in a very fast way. This is called ion torrent. Just the name of the technique is ion torrent. So, yes?
So we don't need to provide a single strand.
Exactly, that's right. No, and don't ask me, I have no idea how this is done actually. But yeah, you have to provide, I guess there are enzymes, right, that just separate the single-stranded. I'm not sure if it's done locally or if you have to provide the single-stranded DNA. So good point though, because from now on, I'm gonna talk about as if we have no clue where this data came from, right? We're just gonna focus on the data and what we can do with it. So to give you a few just ideas of numbers, and I think Eric already mentioned this, the human genome contains 3 billion base pairs. So that's really huge. But in fact, a lot of the DNA is not encoding proteins. It's only 1.2% of the protein, of the DNA in humans that code for proteins. And you might have heard this term, open reading frame, is what is contained between these 3 triplets and will be coding for a protein. So this is, I forgot what it's called. This is the codon where it's noticed that there will be a gene, and this is where then the translation from DNA to RNA will be stopped. And all the codons in between then encode RNA, which then encodes a protein. So in the end, it's not too many if you compare to these 3 billion base pairs. In humans, we have about 20 to 25,000 genes, which is not a huge number. And protein bioinformatics is about finding these sequences, comparing them, and understanding how they're important for function and biology. So maybe this is obvious for some of you. Maybe for some of you, this is completely new. One practical application that probably you've heard about because it's been a very kind of famous thing is looking for BRCA genes. And so BRCA stands for breast cancer. And basically, it's known since a long time that some DNA mutations that are that you can find in these genes mean that you're very likely to develop breast cancer, 85%. And actually, you might know that cancer is actually just a class of diseases, and it's usually due to a combination of genetic and environmental factors. So it's kind of difficult, actually, to find mutations that mean cancer for sure, but this is one of those where it's a really high prevalence, right? So if you have this this gene, you are very likely to develop breast cancer. So through sequencing, it's been possible to make genetic tests for screening, and then 2 courses of action can be recommended depending on where you are and who you are. you might be instructed to do frequent mammograms to just detect cancer if it appears soon enough, and then you can treat it. And some people can possibly undergo preventive surgery. And I guess everybody heard that Angelina Jolie decided to do that, and it became part of the big news and so on. So this is a famous example where bioinformatics has become very practically useful for medicine. This is another example of a slightly different type of bioinformatics. And this is exciting because it's a big project that is led by KTH researchers that are located at SciLife Lab. And it's called the Human Protein Atlas. And the idea has been to, with fluorescence microscopy, label where the proteins are found in the cell and decide that way, or determine that way in which type of tissues they are found. And if you combine then this type of information where locally these things are together with the sequence, then it becomes really powerful, and we can learn many things about pathology. So there's slightly different types of bioinformatics compared to what I've been discussing up until now. I want to give you an idea of how much we can do just with data. And I don't mean that physics will become irrelevant, right? It's not that, but having a lot of data is really powerful in terms of understanding biology in combination with physics. Okay, so our paradigm, you remember this table? Looks familiar? We have on the x and y axes the codon. And the way this is read is if you assemble 3 base pairs, you will get a specific amino acid, right? So TTT will encode a phenylalanine. TCT will encode a leucine, and so on and so forth. So obviously, you don't need to learn this. I guess some people know this. Most people don't. Like, I have no idea. But it's useful to just have in mind how these protein sequences are made. Remember, we talked about this stop codon that tells that transcription should stop. So here you have the stop codon with ATA or ATG. Yes? You're able to read this and to decide which amino acid will be encoded in the protein. Is a start codon. Yeah, so this is something I don't know much about. So ATG here says it's a start codon, but I think, oh, I'm sorry, there's another stop codon here. ATG also encodes methionine, so it has to be combined with some other information. Do you know the answer? No. In the RNA, though, because now I'm talking about DNA. So yeah, you start with the first letter at the top. Yeah. So C, for example. Sorry, I should probably take this. So you take C here as letter 1. Then you take T as letter 2. And then T as letter 3. It gives you a serine. Right? C, T, T. So the first 2 letters determine in which box you find yourself. And then the third letter determines which row you should read from. Yes, so this is the genetic code. And if you're in an open reading frame, like we just said, then this gets translated into RNA and transcribed into protein. And what we end up with is this kind of sequence of letters, which is difficult really to gather anything from. But I'll say I apparently impressed some collaborators last time because I work with a specific class of proteins, and I'm able to sometimes, find motifs that are important and tell people, oh, this is, the active site or stuff like that. But mostly, if you're presented with just a sequence like this, there's not much that you can say or do about it. Even if you have memorized, that a lysine will be positively charged, a glycine would be a small amino acid and so on, it's still difficult to say anything about what this protein does, right? What becomes a little bit more manageable is having this kind of view, a structure, because from this, we might be able to say something about function. And I think you've started learning, right, like how fibrils are made or something. So maybe if you're presented with a fibril structure, you'll be able to say, oh, this looks like a fibril. So I expect that it has the function of making kind of long materials. What we're very interested in is relating this to function. So in this case, this is an enzyme, and it should bind some chemical in order to perform a reaction, and it's whether it will bind this chemical depends mostly on the three-dimensional fold. So we have, again, this relationship between sequence, structure, and function. And one thing I want to highlight again, I already mentioned before, and I think you guys hopefully are starting to become familiar with this. If, let's say, there's an amino acid here that is very important for binding this protein, this chemical, and let's say that an increased binding here will lead to better performance of this enzyme, so of the cell that produces this enzyme, then if this increases binding, if a mutation here increases binding, then the protein, the cell will perform better, so it will have more chance to reproduce, so it will have more chance to propagate this gene to the offspring. And this is the mechanism of natural selection. Is that clear for you, the way it works? Okay. So crucial point here. So this is, a little bit stressing the point, sequence, structure, function, and something that people don't talk too much about because it's like in the modern way of thinking about it, we really want to still consider dynamics. So I think this is what we've been stressing in the past weeks with the molecular dynamics simulations particularly, is that yes, the structure is important. It encodes things that will bind, typically, for function, but dynamics matters, right? And this is why we do, for example, molecular dynamics, is to understand how flexibility also is important for function. Okay, so now we go a little bit more into details. So thanks to all these efforts in sequencing and other types of science, there are many public databases that are available to us that we can use. So we'll mention 3 here, or 4. GeneBank is the database that contains all the genome sequences that have been sequenced up to now. And it's huge, but it really contains a lot of stuff that we don't want. Do you understand, based on what I told you before, why it contains many things that we don't want? Who's talking? Yeah. Exactly. So this possibly contains many things that are not genes, because they can be sequences of DNA that are outside of these open reading frames. So, yes? It has everything else that is not genes. But for our purpose, since we're talking about protein bioinformatics, we're interested in the genes. And instead, so these two databases, SwissProt and TREMBL, contain annotated sequences, which means they contain the genes that are known to encode proteins. So it's a much smaller number. I'll come to that on the next slide. And then we have the Protein Data Bank, which contains the coordinates of 3D protein structures. And this is yet a much smaller number, because there are many proteins for which we know the sequence, for which we don't know the structure. So having the structure involves experimental determination of structures, right? Either using X-ray crystallography, cryo-electron microscopy, or NMR, typically. So now look at this. These are the numbers, just to give you an example. So this is kind of old, but the ratio hasn't changed, even though the numbers have changed. gene bank contains this many sequences, everything that was known in 2007. TREMBL and Swiss prot are this blue and this purple bars. And they have slightly different philosophies. So TREMBL is a little bit more automatic, whereas Swiss prot has a person annotating each sequence. So that's why it's much smaller. And then the PDB is yet an order of magnitude fewer sequences, right? So given that I'm interested in protein function, that function is encoded by the structures, I would like to know more structures. But to determine them experimentally is costly and time-consuming and so on. So the idea here will be to try and relate these numbers or these sequences with the function to try and see if we can gather more information from the sequence directly. No? Yes? And to do this, what we can do is recognize that proteins that have similar sequences have also similar structure. Okay, so I already described natural selection. Apparently this is clear for everyone, so you guys realize that random mutations, insertions, or deletions happen, and that they are transmitted to the offspring only if they are beneficial for the cell, typically the bacteria, but also for the cell. Meaning that we evolve from older ancestors, or any organism evolves for older ancestors, and the proteins that come from a common ancestor are called homolog. And there's two different types of homologs, and this is vocabulary that you might hear if you, or read if you read papers on bioinformatics. So if we all came from the same organism, right? Then in, sorry, I'll start with that. If we all came from the same organism, then we'll have similar proteins in different types of species. So these are called orthologs, and usually they perform same or similar function in different organism, which means, you know, a hemoglobin that is found in a chicken or in a whale or in us will be similar in some way. Not identical, but it will be similar. And then there's paralogs, and those are proteins that perform different functions in the same organism, but they came from the same protein. And so usually what happens is that you have gene duplication, which means by accident the DNA gets duplicated, and then one of the DNA will evolve to promote some function, and the other DNA will evolve to promote some other function. So they still look similar, but they will have a slightly different function. Usually, though, the functions are related. So something that like ion channels, for example, that let through sodium or potassium, they are built in a similar way, and they will not eventually become a fibril, right? It's too different, so they usually have related function. Sometimes, you know, slightly different, but usually similar. So here we have myoglobin from 9 different species. Just to check whether you guys were sleeping on the previous slide, are those paralogs or orthologs? Orthologs, exactly, because they fulfill the same function, but in different species. So this is human, I forgot, this is dog, whale, turkey, many things. And I should say this is a small piece of the protein sequence, right? But this is meant to highlight similarities and differences between these proteins. So if we look here, for example, the green amino acids are completely conserved across all these 9 species. The yellow and white are similar, and then there are some places that really diverge. So I think these blue ones, for example, are outliers. And from this, you can derive a consensus sequence. So if you focus on this column, for example, it's mostly an asparagine, N, in Turkey and, I forgot what PN is. Pony, I think. Yeah, actually, it's pony. In pony and Turkey, they're slightly different, but still you can gather a consensus sequence. And I'm mentioning this because it will become important when we learn to do sequence alignment in a minute. So on the level of the gene and the protein sequence, these are clearly orthologs, and they look similar. What about structure, though? I'll come to this. This is just a digression, but you guys know how we measure differences between structures, because we did this in the lab. Usually we use this RMSD, root mean square displacement. So remember the formula, the difference between structure A and structure B. You take the difference in Cartesian coordinates squared, divide this by n, and take all the square root of this, n being the number of particles in your protein. And then the number that you get from this is a good indication of similarity between structures. So this is quite central concept. Here we have the RMSD of the backbone atom of the core of a protein as a function of the percent of identical residues. And what you can see is that there's a very good correlation between these two numbers. So if you have 75% of residues that are identical in the core, you can see that the RMSD of the backbone atoms will be less than 1 angstrom. So this means that they're very similar. If you have 50% identical residues, you're still around 1 angstrom. But then when you go to lower numbers, below 25, you're already at a much higher number. Okay? So what does this tell me? If I have a sequence and I'm interested in the structure, but I don't have an experimental structure, if I can find another protein, whether paralog or ortholog, that has a similar sequence and a known structure, then I can infer the structure of this protein that I'm interested in. This is called using homology to infer protein structure, and it's the basis of homology modeling. Make sense? So in order to make a good three-dimensional model, either I have a structure, or if I don't, but I can find what happened. But if I don't, then I can probably make a model. So this is an example of two proteins that have 30% sequence similarity. And you can see by superimposing them that they're not identical, but they're very similar, especially in the ordered regions, these helices and these beta sheets. And so they only have 30% sequence similarity, but the structural similarity is very high, right? Or I don't know about very high, but it's quite high. So 30% was here. And the idea was that we would have an RMSD deviation that would be around 1.5 angstrom. And remember what you learned in the lab, 1.5 angstrom was something that was small, right, on the backbone atoms. Why is it that the structural similarity is higher than the sequence similarity? Can you guess? Yes? Yeah, that's right. But why? Has to do with polarity. Mm-hmm.
So if one polar amino acid was exchanged for another polar amino acid, they would have the.
Yes.
It would be a big difference if a non-polar one.
Yeah. So what you're saying is true. In fact, I think this has been mentioned, and I'll talk about it also in the next lecture. There are not so many different folds that are possible. So nature really has, really recycles structures. And it is related to the chemical nature of amino acids and the fact that there are only 20. But really, it's about recycling. So once nature has found a fold, it will be very difficult to find a different fold through natural selection. But it's kind of easy to evolve within a similar fold. And this is why the structural similarity is higher than the sequence similarity. So if we think about how easy it is to then detect similarity, there will be this kind of diagram with 3 zones. If we're above 40%, then it's very easy to detect sequence identity. If we're below 20%, then it's very difficult to detect it. And in between these 2 regions, it's quite hard. But this zone keeps getting bigger because we know more and more sequences. So how do we detect homology from sequence similarity? Well, we're going to have to align the sequences. So this is your example for today. Imagine you have these 2 small sequences. The question is, how do you align them? So do you just align them? How long is this? 11. So do you just align all 11 one-to-one, or is there a better way to align them? And going to this, maybe you can tell already that this is a better alignment because you're aligning K to K, F to F, L to L, F to F. Yes? You see that this is a better alignment than this one, but it involved placing so-called gaps. So to align, you're allowed basically to introduce gaps or, in other words, remove amino acids from the other sequences. So that would be called insertion or deletion, respectively. You're allowed also to place the ends slightly mismatched. But how do you do this? OK, just highlighting insertion, mismatch, and match as vocabulary. Match means same. Mismatch means different. Insertion means that on one of the sequences, I will have gaps that correspond to positions on my other sequence. Yes, sorry.
Are there any amino acids that we know or that we want to prioritize to match?
We're getting there. So let's just let me follow the whole kind of process. I know where you're going, but I don't want to confuse people, but I'm highlighting that what I call a match here is exactly the same amino acid for the moment. So this is about identity. So C matches C, K matches K, and R doesn't match anything. So for the moment, we're talking about identity, but we're coming to your point in very shortly, actually. So to detect how we should build this, now that this is done, you can notice that this is a better alignment than that. But to do this, we can do something quite simple, which is called a dot plot. So you write your one sequence in the x-axis, you write your second sequence on the y-axis, and then you just put a dot in the matrix, or a cross, whatever, when there is a match. So C matches C, K matches K. You notice that since there are 2 L's in this sequence, and in this one, that I now have 2 dots, L matches L here, and L matches L there. But L also matches here and there, right? And same for phenylalanine here. You have 1 phenylalanine that matches 2 phenylalanine and vice versa. So in most of the cases, because this is a very short sequence, I only have 1 match, but I might have 2. And then what we do from this is that we will delete the points in the matrix that are in less than 3 on the diagonal. So it will look like this, right? This was an isolated point, and this also. So we got rid of them. We, again, remove all hits that were shorter than 3 positions. And with this, now I have my alignment. So I'm saying I want to align CKF here with CKF here. This is this. I want to align LFG DELR with LFG DEL. sorry, no R on here. And this basically gives me this alignment rather than this one. Yeah? Simple. So this is a dot plot. Now the problem with this kind of dot plots is that if you take a real sequence, now this gets much more messy. So this is hemoglobin, chain A versus chain alpha versus beta. You can see that these sequences are much longer than what I was just talking about, right? And then you have this kind of map. And it's not so easy to interpret. There are many false hits, and it's quite hard to quantify. So basically, this says you have to zoom here to see that these are not really continuous rows. But you might not be sure if you should align this part or this part. So this is where we go to more sophisticated methods. And that's what you were talking about. And instead of looking only at identity, now we're going to look at similarity. And the idea here is that via evolution, it's easier, it will be more favorable to, let's say, replace an arginine with a lysine, because they're both positively charged, than an arginine with a leucine, which is a bulky hydrophobic residue. So there are amino acids that have more similar physical properties, and evolution or biology can use them in a similar way. And this is we'll then measure similarity and use this when we will pair our residues and make an alignment. I talked already about the fact that there's also the possibility of inserting, of having insertions or deletions. And this is the key of what we'll do. We will try to maximize similarity without adding too many gaps, because we know that it's costly for evolution to introduce gaps. So the similarity measure will be done using these matrices, and I'll come to that in a second. I think I've already talked about this. We want to conserve amino acids. That would be very good. We will allow similar amino acids to be substituted, but also we will avoid significantly different amino acids. And we have 20 amino acids in the alphabet, right? And so this is the matrix of Blossom 62. Again, something that you don't need to notice, but to memorize, I mean. But it's quite interesting to look into the details of these numbers. So I'm like, this is the score that you would get for replacing an alanine by an alanine. This gives you a score of 4. Replacing an alanine by a proline costs you -1, because alanine and proline are quite different, right? Remember, proline was the one that had the ring structure in its backbone, whereas alanine is a relatively small amino acid. Alanine likes to sit in helices, prolines really don't. So there's a cost when you make this alignment to substituting your alanine by your proline. You get the idea? Remember what I was telling you about lysine to arginine? So lysine is K, to arginine is R. Both are positively charged. You get a +2, which is quite good score. Okay? Maybe one amino acid or one number that stands out in this matrix is this substitution W by W. See a big fat 11 here? What is W? Tryptophan? No? Okay, just another amino acid to remember maybe. This is the biggest of all. It contains 2 rings, and it's very distinct compared to the other ones. It has interesting properties because it's big, hydrophobic, but has some polar moiety, so it does really intriguing things, actually. And so it's very difficult to replace by anything else. So this is why on this row, all of these numbers basically are negative, except there's tyrosine and phenylalanine here that have slightly positive numbers because they're also aromatic. But basically, if you have a tryptophan and you keep it, then it's a big score, just because nature can't do anything else with it than keep it in its place. Yes?
So when you say change a W for a W, you mean keep it?
Yeah.
Oh, and what's the unit? Or is there
There's no unit, so here we come to that. It's good you should ask every question, because it just drives me forward. So the problem with this is that it's based on data, so it's not based on physics. So there's often no kind of unit associated with it. It's just a scheme through which we score things. So we could use any really score, and actually if you go into softwares that allow to do this, they will give you default values, but you can always vary the scores that you're using. So I showed you this Blossom 62 matrix, but first there are others. So they have a slightly different way of scoring. And then you need also to give the number that you want to assign to a gap. And usually there's two parameters here. There's gap creation and gap extension. And I think the default values in cluster W is like, it costs 10 to insert a gap, but it costs, so minus 10, and it costs minus 1 to extend. So it's very expensive to put in a gap, but it's not that difficult, it's not that costly to insert a gap. And then basically you just compare your two alignments, and the one that has the highest is the best that you have. So this is an example that doesn't actually use the similarity matrix. It just scores matches, mismatches, and gaps. And by looking at this or that, it might not be obvious without the scoring what is the best. But if you just add, so 3 times, I think this was 9, and 8 mismatches, no gaps, you get a score of 27 minus 8, that was 19. Whereas here you have many matches, but you do need to pay the price for a gap. And in that scheme, the gap is not very expensive compared to the mismatch, so it's a much higher score to use this second alignment. You get it? How they're built, how they're scored? Good. So before we take a break, I'll just say a few words about how we can improve this. We have so far used this kind of generic way of scoring similarities that was mostly based on chemical and physical properties. But we can exploit evolutionary information, and this is what's been used nowadays, actually, almost always. So if we start with a seed alignment, let's say I provide you with these 9 sequences that are already well aligned, then adding more sequences becomes easier because we can build a position-specific scoring matrix. And notice the difference from before where I had sequence against sequence. Here I have sequence against the position in the multiple sequence alignment. So this actually doesn't match the previous example, but let's say at position 1 in my alignment, I just look at the alignment that I have, and I notice that it's very often a Y tyrosine, and it's never a proline. So then I give -4 for proline at this position and a +5 for tyrosine at this position. And I do the same at position 2, position 3, and so on and so forth. And now I have a position-specific scoring matrix, which means that for this family, I have a specific scoring, contrary to the generic scoring that we had with the Blossom 62. Does that make sense? Yes? So then we can search an entire database looking for good hits and add them to this list. And maybe eventually we find one for which there's a structure. So this means that if I don't have the structure of the human, I'm going to be able to use the homology modeling to define the structure of the human. Yes?
In the next slide, I don't understand completely what you meant with if you don't find many proline.
So that would be, let's say, at this example. So here I have position 1, 2, 3, 4, 5, 6. I want to find a good one. 7. So the column 7, since it's very often an E, it will have a very positive score for E, a slightly positive score for D because it's found once, and a negative score for everything else.
So that's when you compare it to other homologs.
So that's when you have a sequence alignment to start from, right? So on this, where can I find one? On this position, for example, I don't have a lot of information, so that wouldn't be so informative. But for the later positions, here, for example, from this example, I know that it's a V 100%. So I will score very low any other thing. So in that way, we can find sequences that resemble those based on the probabilities of finding these different amino acids in there. So this is really based on data, whereas the previous one was based on physics and chemistry. Yeah. And so to conclude this part, basically, if you now consider, this is an example from a bacterium that contains 4,800 proteins. If, and let's say we don't have the structure of any of these proteins in that specific case, if we were to use only the sequence identity, we would be able to predict the structure of about 10% of the proteins. If we use not sequence identity, sequence similarity, so using the Blossom matrix and homology modeling, we would be able to get the fold of about 10%. Now, if we use this position-specific matrix and we make predictions on the structure using the profile, we can get 3 or 4 times the number. And I didn't have time to go through HMM, hidden Markov models, but in this kind of technique, instead of using only the probability at each position, you also use the probability of having a sequence. Then you can even reach higher, and this is the total number of sequences. So with the profile, you get about half, and with the HMM, you can get about 3/4. So this makes it quite powerful to know what proteins will look like in your unknown bacterium. And I will stop here to take a break, and then after we'll talk a bit more how we can predict protein structure in practice. But first, a question.
This question with Luhmann, which table was it?
That was with the PSSM. Yes, exactly. So let's say this was with the Blossom type, this was with PSSM, this was something I didn't explain. Yeah? Questions? No? Okay, see you in 15 minutes then.